We're going to get interrupted again because he has to bring in the cart thing, so just ignore him. Pretend he's in the man behind the curtain. Okay, so what we're going to do today is kind of go back. We did skip this earlier on, mainly because we had what was a snow day earlier, and so I took that lecture away, but we managed to have a little time to cover this. And I find that it's actually necessary because there's many people that think they know what the mitochondria does. There's actually a lot of people who just don't get the basic idea. And so I have students that come to my endocrinology class after having taken biochemistry and they just don't understand what's going on. So I don't want people like that coming out of my class to go to someplace and they don't understand the basics of how energy is produced. Okay, so that's why we're going to do it. And so we start with mitochondria, and let's actually go to the outline. Mitochondria is interesting because of the way it evolved from a separate organism. So the endosymbiotic theory was developed by Lynn Margulis at Boston University. She actually gave us a talk about 6 or 7 years ago. She was a plenary speaker about how this symbiotic relationship evolved between a bacterium that was aerobic that could actually perform oxidative phosphorylation and an anaerobic prokaryotic creature that had the first nucleus. So those two had to come together, and the idea was, as actually shown in this slide, so both mitochondria and chloroplasts probably evolved from bacteria that were engulfed and enslaved by a anaerobic eukaryotic cell, but it had a nucleus. So it had an advantage already because its DNA was separate from the stuff that goes on in the cytoplasm, whereas the poor bacteria had no nucleus and unprotected DNA, but it could do something that the eukaryotic cell couldn't do, and that is make ATP from a proton gradient. So this engulfed that, and so you ended up with an organism that has a nucleus, which is the eukaryotic cell, and this is the engulfed bacterium, and it has a double membrane structure, just like a gram-negative bacteria. What other similarities are there between this structure, the mitochondria, and prokaryotic organisms? What can you think of right off the hand that's very similar?
Like other gram-negative bacteria, it has membranes.
Two membranes. The outer one is fairly permeable, and that place in between is called periplasmic space. And so it can exclude some things, but there's a lot of stuff going on in the intermitochondrial membrane. That's the plasma membrane of a bacterium. What other similarities are there between, let's say, the DNA of a mitochondria? Mitochondria have DNA, right? And so do chloroplasts. What does it look like? Anybody know? Yeah.
Circular genome. It also doesn't have a bacterial-sized ribosome as well.
It has, exactly. So it's a circular piece of DNA. Ours are not circular. We have chromosomes, right? And as he mentioned, the ribosomal subunits are small, like a ribosomal, or like a bacterial ribosome. So the ribosome is the same. The genes are circular. What's happened, though, is over time, is like any parasite, what happens is if you're living inside another organism and the host can take over some of your jobs, which is, so the host here is a eukaryotic cell. What was the advantage to the bacterium? Well, it had a protective cell cover, okay, so for protection, and it provided that bacterium with a source of nutrients. It didn't have to do that anymore. So it actually lost genes. The genome got smaller, and the functions were replaced by the genes. So the mitochondrial genes got smaller, and then the nucleus produced nuclear genes that took over those functions. And so that mitochondria now cannot function independently because it's lost the ability to make stuff that it needs. It's given that job over to the eukaryotic nucleus. Okay, so any other similarities? There's some weird lipids that are only made in bacteria, and they're also made in the mitochondria. There's a few other things, but it's really clear that this had a prokaryotic ancestor. Okay,
They
now
replicate separately from your cell, right?
pardon me?
They replicate separately from your cell, right?
In here, no, they're actually replicating inside by fission. Oh, that's the other similarity, is that mitochondria self-replicate, and they do it not by mitosis or cytokinesis, but they cut themselves in two, which is what bacteria do. Yeah,
Where is the DNA exactly located?
in the matrix. And that's good, because the compartmentalization of mitochondria are complicated. And so there's the matrix, that's our cytoplasm, that would be their cytoplasm. The inner mitochondrial membrane is their plasma membrane. The outer one is just sort of a size exclusion device, okay, a protective device. But it's got porins, as you know, that's a beta barrel motif, and it excludes things that are larger than 5,000. So nutrients and pyruvate and stuff like it, there's no pH difference between the eukaryotic cytoplasm and the intramembrane space. It's all pH 7. But the cytoplasm, or the matrix, which is the cytoplasm of that mitochondria, is tenfold higher in pH. So that means it's got a tenfold pH gradient, lower number of protons than the intermembrane space. So what creates that pH gradient? That's the key to understanding energy. So any questions?
Why is this only inherited from the mother?
Because the sperm is just, he has one job. He's wiggly and he has a head full of DNA, and that's all he wants. And so usually he does not deliver. They have a couple of mitochondria. That's his speedboat. That's how he propels the flagellum, right? But those mitochondria often do not make it into the fetal, into the egg. It's the maternal, that's why the egg needs to be so big, because it's got to do everything mostly. All you got from the sperm is some DNA.
Is the mitochondria from the sperm made inside the egg? I know that I've heard that the mitochondria in the sperm are actually like specialized in like a different shape or something like that.
Right.
Does that actually function normally, or would they just not replicate, or is there anything showing that?
That does happen, and there's no reason why that wouldn't contribute to normal mitochondria. Because once they're in the cytoplasm, it's just everybody's a mitochondria that's a mitochondria. I don't know if there's problems with that. It would sure screw up the genetic analysis if you were depending on the fact that it was only maternally inherited, which is most of the time, but biology always finds a way to screw things up. Okay. All right, so we have this. There's actually, let me see if I can play this. I guess I didn't bring it. There's a pretty good, if you have that DVD, there's a thing called a tomogram, and we'll look at it. You use a high voltage electron microscope to make a three-dimensional picture of that thing. What's wrong with this? The problem is, to get electron micrographs like this, you have to treat that cell with really nasty things, really nasty acids, protein chelators, heavy metals. It's bizarre. And what happens is there's shrinkage artifacts. So this thing is never going to look like this. It actually looks like a worm. It's really long. But in response to those fixatives, they shrink up. We can see it, but that's not what it looks like. But you can see a little bit. You can see that there's an outer membrane, there's an inner membrane, there's the intermembrane space in between here. And then there's the classic enfoldings, which are called cristae. Okay? And that gives it more surface area to pack on dozens and dozens of proteins. There is only standing room, only in that membrane, for all the proteins that are needed. Electron transport chain, ATP synthase, all the transporters to get the nutrients into the matrix, lots of stuff going on there. So it's packed with protein. Okay. So let's see if we have a better picture. So this is this three-dimensional tomogram that I was talking about. If you play that on your DVD, it will actually rotate for you to show you what the three-dimensional structure of those little enfoldings look like. Okay. And so that can be sort of informative. And there's also some, if you go on the web, there's lots of stuff about the mitochondria. So they'll show you in real time mitochondria that have been stained with green fluorescent protein. And so you'll see these things wiggling around. But you notice there's gaps here. They're not going into the nucleus, right? The mitochondria are way too big to go through the nuclear pore, so they don't go in there. And there's areas here where they're not going, and that's because they travel on microtubules. There are kinesins and dyneins that are moving, that are carrying these things. Okay, so they're moving, but they're being escorted down the highway to go to a particular place. Now, why is the location of the mitochondria important? Why would it be important? Think about that question based on what it produced. So it's making ATP, right?
Wouldn't you want the ATP to come out where it's used?
Exactly. Because this is a diffusion problem, right? Diffusion is fast at these distances, but it is a problem, okay, if you're using lots of ATP. So I'll show you a couple of cases here in a moment where that location is very important. And these mitochondria are targeted and they are transported there by molecular motors on the cytoskeleton that we just talked about. Okay, that doesn't work. Okay, so this is a good example. Skeletal muscle. It's just crammed full of contractile machinery, and as you know, the nucleotide that is being split on the myosin head is ATP, right? Something has to supply that ATP or else you go into rigor. And you know what rigor is now. It's when the myosin head sticks to the actin. It has to have ATP to let go and then move forward towards the plus end of the actin filament. Well, all that's powered by these guys. So these things are transported in between the sarcomeres. And so the ATP only has to diffuse varies a few microns, a couple microns, maybe half a micron, to get to where it's used, which is right there. Okay. And so that's what you have these. And then another place, of course, on the axoneme of a flagellum, sperm flagellum, that mitochondria are wrapped around these things to provide that little propeller with ATP to make it work. Okay. And also you can think of, let's think of, I won't draw it, but think of Bart Simpson, you know, the enterocyte with his little spiky hair facing the lumen, and then on the basolateral side is facing the blood. Okay, so they're different, there's different transporters on the apical and basolateral membrane. What's on the basolateral membrane that uses ATP? It should take you about 2 seconds. Anybody else? Sodium glucose, simple. Think about how sodium gets across. That was on the test, by the way. How does sodium get across from the lumen into the bloodstream? By mechanisms that we know. So it gets across Bart Simpson's haircut using SGLT1. Okay, so 2 sodiums and 1 glucose comes in. The glucose is supposed to go out unless it's blocked by cytokeratin like it was in the test. Out through GLUT2, the sodium exits via the sodium pump, which is splitting ATP. Where are you going to locate your mitochondria? Where it's used, and it's being used where? The basolateral membrane. So you see all these little mitochondria hanging out down near the basolateral membrane, but they're not up there because it's not running off of ATP. All those transporters at the top are using the sodium gradient and not ATP. Is the sodium gradient directly caused by ATP hydrolysis? Absolutely. So it is the same energy source, which is ATP ultimately, but the sodium gradient is a form of free energy. So that's a really direct, simple concept that everybody needs to understand. Did someone have a question? Okay. All right, so here's the idea. You can either get energy from sunlight, which we don't do very well. We tend to just darken. Okay. We eat stuff, and what you're going to do is break. Your digestive system has the lovely job of breaking all those goodies down into monomers. And that's so that those monomers can be transported by Bart Simpson into your bloodstream. And then those nutrients have to go into your body. And taking an example, we'll focus mainly or almost exclusively on glucose. So you're going to digest all those starch polymers, chop them up into glucose, and then send them into the bloodstream. Cells take them up through the GLUT family, mostly. Okay. And then what you're going to do is you're going to extract high energy electrons from that glucose molecule. And you're going to do it in a couple of different ways, with oxygen and without oxygen. So when you're doing it without oxygen, that's the really most fundamental way that an anaerobic creature, our ancestor, the eukaryotic thing that had no mitochondria, had to do it. Glycolysis is the process of anaerobic oxidation of glucose. It's really fast, that's one advantage. Its disadvantage, it only produces 2 net ATP for every glucose molecule broken down. That's sad. That's not going to do very well for an organism as complex as we are. So we rely on our mitochondria bacterial friend to produce ATP much faster and much more potently. Okay, so what we'll do is we'll ask the mitochondria to produce a proton gradient, and then we're going to, so that's a stored form of energy like the sodium gradient. It's the same stuff. Okay? If you can create that gradient and then run it through the ATP synthase, you already know what that is. It's the FOF1 ATP synthase, ATPase. But it's not splitting ATP, you're running it backwards to create ATP. So you take any pump and you run it backwards. Can you do that to the sodium pump? Can you make ATP if you set the sodium and potassium gradients correctly? Yeah, you can. You can run it backwards and make ATP. But normally it runs forwards, it splits ATP and ejects sodium-3 into the extracellular space and takes 2 Ks in. Well, this thing can be run in either direction, and I can prove it to you. Okay. So, all right. So that's the whole idea. But it's a little tricky how to generate this thing and then get it to go through here. Okay. We can solve that problem. Okay. So what is this? Take a moment to commit this to memory. This is, you notice that there's all these metabolic pathways. What is really central to all of this stuff? It's this thing. So this is glycolysis, and that's the TCA cycle. Okay. There are proponents of the metabolism first hypothesis. So there used to be, when life was formed, DNA was the thing that came first. If you didn't have DNA, you couldn't be a cell. Okay? Well, really, you can't be a cell if you don't have a membrane. But you need to have a membrane and the DNA in there something. And then people said, that's kind of difficult because DNA is not catalytic. All it does is just store information. And so a number of people evolved to suggest that RNA came first because RNA can store information, can carry information, and it's catalytic because it has secondary and tertiary structure. So it's got shape, it's got a surface to it that DNA doesn't. DNA has some characteristics, but it's boring compared to RNA, which is catalytic. What do you call catalytic RNA? You have enzymes and you have ribozymes, okay? It's catalytic, so it can do a lot of things. But then there's a more recent school of thought that says, if you could construct this metabolic pathway, what you can do is you can actually make ATP, so you can get energy out of this thing. So this is the core. So this is glycolysis, and this is the citric acid cycle. So you can create energy. High energy electrons can be captured, so you can power things. And then if you run it backwards, you can make amino acids, which you need to make protein. You can make nucleotides, which you need to make RNA and DNA. If you have this, you can make everything. So there are a lot of proponents that this thing came first. So it's not trivial that we study this. Now, do we have to memorize all this crap? If you want to do that, please go take a biochemistry course. Okay? Have lots of fun doing that. But I have a lot of students that come out of biochemistry that don't know. They can tell me every one of those structures and they can recognize it on a flashcard, but they don't know how it fits together. Okay? So I don't want you to be like that. Okay, so we're not going to memorize all the steps of glycolysis. We're going to ask the question, what's important that comes out of this thing? Okay? So we start off with a 6-carbon glucose. And what's going to happen is, right off the bat, we actually have to use energy instead of producing it. So it actually takes an investment of 2 ATP just to get to this state, this fructose 1,6-bisphosphate, okay? And then you're going to cut it into 2 pieces. So from then, every step is just 2 times what's happening on each one of these. So you get 2 pieces, and you're going to, on this side, you're going to make, you're going to get 1 NAD. I call this NAD, okay? And we'll talk about the NAD fat burners. So this is NAD, it's nicotinamide adenine dinucleotide, okay? And it comes in 2 forms, an oxidized and a reduced form, okay? When it's carrying electrons, it's got a little H after it. So that's, you can always say NADH, that's reduced. It's carrying electrons. And what that means is it's carrying energy. And then when it gives up those electrons, it becomes oxidized, and the oxidized form of NADH is NAD plus. So that it shuttles between those 2 states, NAD plus and NADH. Okay. And it's really important that you understand which of those 2 forms carries the electrons and which one doesn't. And I'm going to show you a picture of NAD because his family wants you to see that. Okay. So you're producing this high energy, but you're only getting one of these pairs of electrons. Actually, you get a second pair because both sides of this is split. So you're going to produce 2 molecules of pyruvate. So you'll get 2 NADHs, and you'll get actually 4 ATP, but you put in 2 to begin with. So what's the net output of this whole thing? 2 ATP and 2 NADH. Okay. Now you've got to you have to extract the energy from NADH. You can't do it in the cytoplasm. You have to give those electrons over to the mitochondria. How are you going to do that? Well, it's too big. It'll go through a porin, but it can't get across the inner mitochondrial membrane. This is too large. It is mobile. It will go through there. Okay. So what you do is you use a couple of electron shuttles. NADH never goes across the inner mitochondrial membrane. It hands them off to NAD on the other side of the membrane. Those are called the electron shuttles. Okay. And there's a couple of them. Okay. There's the malate shuttle. Okay. And what that does, let's see, do we have a picture of that? Okay, so this is what we're talking about. This is the problem. That cannot diffuse across here. Let me see if I have a picture of it. No. Okay, I'll draw it for you. So, oh, they took my thing away. Let's see if I have a way. Without turning this off, I hope this is what is that big? Here, I'll tell you what. Blind somebody that tries to sneak in here. There's just dozens of people wanting to come in. They hear the word mitochondria, everybody gets excited. All right, so this is the mitochondria, and the outer, this is out for that cytoplasm, and this is the matrix. Okay, and this is the space, and this is the membrane that's the problem. Okay, so what we're going to do is we'll just ignore this outer membrane, so just pretend it's not there. So we want NADH in the reduced form. It's carrying the load of energy. And what we're going to do is hand its electrons off into what's called the malate shuttle. And the malate shuttle is not simple, but we're going to represent it by a black box. Okay? And so the point is NADH is going to hand off his electrons to oxidize NAD on the other side. Remember, this is the one that wants the electrons. This is the one that has the electrons, and they're going to shake hands, and those electrons are going to come through. NAD is going to pick them up and become NADH in the matrix. What a sleight of hand trick. Okay? And then what happens is you get another bonus because what you're producing on the cytosol is NAD plus. Why must you have NAD plus? You have to have NADH give away his electrons because of why? If you build up this huge cytoplasmic concentration of NADH, glycolysis will stop. you have to have an electron receiver, okay? And so the only thing that can receive it is NAD+. So you're regenerating the electron receiver, okay, acceptor. If you don't produce NAD+, glycolysis stops. Okay? So you get a twofer. You get these electrons transferred where you can use them, as I'll show you, and you get to regenerate NAD. So that's really important. Now, there is another electron carrier, and that one is a little strange. This is FAD, flavin adenine dinucleotide. Okay, and it actually carries, when it picks up 2 electrons, it always picks up 2 protons, and so it carries 2 hydrogen atoms. That's how it carries its electrons. NAD, I always like to say, NAD, these 2 guys are brothers, NAD and FAD brothers. NAD is a Texas Tech student, so he's very mobile and very useful. FAD, I think he went to A&M. Because he can't get around, he's made out of rubber, rubbery, and he has to stay associated with the intermitochondrial membrane. Okay, and he's a little slow, he doesn't move. Okay, so he can't go out and grab electrons. So you have to bring it to him using something called the glycerol phosphate shuttle. Okay, and it's pretty much the same story. What happens is the Texas Tech guy is fast, so he shoots him a couple of electrons just to make him feel better. Now that he's left the Big 12, and what FAD does is he will take those electrons and go from being FAD, which is the oxidized state, and that will convert him to. He has to stay in the membrane, by the way. So he becomes FADH2. So that's his reduced state. Now both of those guys can then deliver their electrons to Christy, which gives her a really bad case of PMF. Okay? And we'll talk about how that happens. All right, questions about why you need these electron carriers? So far, it doesn't seem like, who cares? We're not, we got very little ATP out of this thing, but we're going to get some payback for delivering those protons. Okay. So now our focus is going to shift into the matrix. What's the free carbon thing we get out of glycolysis? Because we're not done with that yet. Pyruvate, right? And it's acidic. Okay, so it's going to go into the matrix using a pyruvate proton synport. So it's electroneutral because pyruvate is negative. The proton goes with it. And let's just draw it. So we have this pyruvate is a carboxyl group, so it's negative, right? And so it goes right through the boron, doesn't have any problem. And then there's a symport takes the pyruvate in there, and then along with a proton. Okay, and that's good because you have a really hard time shoving a negative anion into the matrix. It's very negative in there. You might know what the membrane potential is inside of mitochondria? Remember, this is a bacteria. What is the cytoplasmic matrix potential relative to the outside? Negative 160 millimoles. It's not 60. It's not, your neurons, when they're really impressed with themselves, go down to minus 90. This is, the VM on the inside is minus 160 millivolts. You're not going to push pyruvate by itself in there. So what you do is you fool it by taking a buddy that's positively charged, and now it's electroneutral, pyruvate slips right in. Why? Because the chemical gradient is high on the outside and low on the inside. You're using pyruvate and you're producing pyruvate on the outside, so it's going to just go right in. Good. So now we got everybody we need. We got a 3-carbon pyruvate, and we're going to pick it apart. Right? Plus, we've got a couple. We have at least one. We have 2 NAD, so those 2 NAD have been regenerated into NADH on the matrix side, and then you have there was no FAD produced, so we're going to use. But FAD gets handed off to here in a moment. All right. So, what do we do with that? Okay, so this is the overall plan. And that is, we're going to take the pyruvate, we're going to run it through the citric acid cycle, run those carbons, and we're going to pluck them off one by one. And it's a 3-carbon molecule, so how many CO2s are we going to get out of this? If we completely oxidize those carbons, we puff them out as CO2. And the first one is puffed out right here. So pyruvate dehydrogenase that we talked about is this amazing multi-complex, multi-protein complex, chews on pyruvate and turns it into a 2-carbon unit that's going to feed into the citric acid cycle. And then you blow off the first CO2. That's because you've taken 1 carbon and done everything you can. If you cannot oxidize it further if it's at CO2, right? And so you huff that out, okay? And then this is going to go through, these 2 carbons are going to go through here, and we're going to blow 2 more CO2s off, because that's all you have left, and we're going to capture a bunch of NAD, 1 FAD, actually 3 NADs, 1 FAD, and then 1 GTP. And then GTP equals ATP, that you can convert them back and forth. So you're going to get some really good stuff. And then we're going to take our NAD and FAD brothers, and they're going to hand off their high-energy electrons to the electron transport chain, which is in the inner mitochondrial membrane. Okay? And when we do that, what we're going to do is produce Christie's PMF problem, proton motive force. So that's where the energy is going to be stored temporarily until we use it to make ATP. So that's the plan. That's the overall plan. You got to be able to know what the force looks like before you look at the trees. Okay? And guess what? We can actually use fat. A lot of us can stand to burn more fat and less glucose, but you can take fatty acid and you can turn them into acetyl-CoA. That's called beta fatty acid oxidation. And so you can produce the same thing, acetyl-CoA, that you can run through the TCA cycle. So fats are good too. All right, now, we're not going to memorize all these steps. What we are going to know is that we're going to slowly break down these 2 carbons in very small discrete steps so we can harness the energy as NAD, NAD, and NAD. So we get 3 NADs out here. We get 1, sort of crippled A&M student that comes out of here. And so all those NADs and FAD are going to deliver their electrons to the inner mitochondrial membrane. Who are they going to hand it to? The electron transport chain. Okay. So that's where the energy, and this thing can be converted to ATP, so it's pretty much equal to that. And then you can see where the carbons are blown off, and then you expire these. Right? So now we've blown off all 3 molecules of pyruvate have been completely oxidized and stripped of the energy. Now we've got to recreate it in a usable form. Okay. That's the goal. Okay. So that's why the TCA cycle is right at the core of that metabolic pathway. This is just a quick, we don't want to memorize the structure of NAD and FAD. It's to make you understand that it's important to know that that's the reduced state that's carrying electrons, and this is the oxidized state that's giving them up. It can pick up another pair. Okay. That's what you need to get out here. And it just shows you that you're going to carry these electrons in different ways, and then at the end of the day, what you're going to have is NAD is oxidized, and that electron pair that was carried on this really weird way is going to be given up to the electron transport chain. This is fairly unstable. So NAD wants to run around and give it up. Give it up to either the shuttles, if he's in the cytosol, or to the electron transport chain if he's in the matrix. So that's his job. Okay, and then there's his slower brother. Okay, and FAD with no pluses or anything that's the oxidized state, and when it picks up electrons, it picks up 2 protons, hence the term H2. So it carries its electrons in a different way. And notice it's very rubbery. Okay, it's got all this. That's why it loves to just sit in the inner mitochondrial membrane. It's actually buried in there. So it cannot move around. It can't go into the matrix. It can't go into the cytosol. Okay, but it's just an electron carrier. Okay, now this gets a little bit more detailed. So if NAD and FAD take their electrons to the electron transport chain, what's going to happen there that's important? Well, those electrons are going to be given up to different complexes. And we're not going to memorize their names. We're going to call them complex 1, 2, 3, and 4. Okay? And so we give it up there, and those electrons are going to be passed sequentially by very clear method of who you have to go in order, okay? And there's a reason for that, and you need to know what the order is and why it's important. But as those electrons move through this chain, you're squirting out protons. Okay? You're taking protons from the matrix and pushing them into the intramembrane space, which, guess what, equilibrates with the cytoplasm. Okay? So the cytoplasmic pH is the same as the intermembrane space, which is more acidic than the matrix. In fact, because of this pushing, this electron push, I'm saying electrons, this proton push, we're creating one hell of a membrane potential. That's why the inside is negative 160 millivolts. You're moving these protons without any counter ion, correct? And every time you do that, you hyperpolarize the cytoplasm of that bacteria, which in this case is the matrix. So that is the origin. You need to know that. That's the origin of the negative 160 millivolts. And it's lots of stored energy. So the electrical potential is most of the energy that's in the proton motive force. It's not the chemical difference. What's the chemical difference? Well, this is, let you figure that out. This is a pH of 7, like it is in the cytosol. That's a good round number. What's the pH of this? It's 10-fold more protons out here than in here. If this is pH 7 and this is 10-fold less protons, the pH is going to be higher or lower? Higher. So it's pH 8. That's a really easy way to remember that. pH 7, pH 8. So there's only a 10-fold chemical potential. So we're not going to get most of our energy, but we're going to get about 60 millivolts in energy from just the chemical potential. Because we're actually moving protons in concentration and concentrating them out here. And then to end the story, all we have to do is understand how we can run them through ATPs. And that's going to actually spin a turbine and produce ATP. So let's take a break here, and then we'll come back and finish the story. Okay, why don't we get started again? So as soon as I have those test scores, I'm going to put them up. I'll send you a Piazza. I expect that's going to be this weekend. Okay. I still have a person that hasn't taken the test. That's why you can't get the test back. But if you're really confused about a particular question, you can come talk to me about it, and I can give you the information you use to answer that question. Okay. So if you want to do that, that's fine. But that's why I can't give you the test back. I can give you your grades though, and that will become because you need to. Be encouraged to study for the final exam. Definitely. So in two ways. If you did really bad, you're going to have to work really hard. And if you did pretty good, you know exactly how well you have to deal with the final. So it's going to be informative. There's a lot of, half your grade is on that final exam. That's why you've got all this extra time. We're not going to do anything new on Monday. We're going to do a couple things. You need to come because I'm going to take attendance. And I want you to bring questions, and it'll be a review session, no new material. Okay. We might have to finish up something, but it'll be very, very minor. Okay. And then, that's what's due. That's no new material. Thursday's posters, no new material. The following Tuesday, no new material. But don't use that time to study for other stuff. Use it to study for this class. Okay. You're not taking anything else. But use your time wisely. Because when I delayed the test, I think what everybody did was just give them 4 more days to wait. And that's not going to cut it. The nice thing about the final is there'll be about 60 questions. You'll have all the time in the world. So they're only going to be worth, 2 point something. It's not going to be letter grade. That's really useful. And it's stuff that you've had over and over, plus this little bit of new material. So it's very doable, and if you nail that, you can change your grade a lot. I've had people go from B to B, and I've had people that low C's go to A's. So it happens, and if you do the math, and you do your poster appropriately and all those exercises, you can get there. You can solve for that grade. When you have your test 3 grade, you can solve for what you need on the final to make a certain grade, assuming I'll get that. Pretty good on the poster. I've got all those 50 points. It starts to look really good, but don't blow it off by not starting out. Use that extra time. Okay. So, where were we? We have to understand. Can somebody close that? Thanks. How we can deliver these electrons and get ATP out of it. And if we just take NAD and react it with water, we get an explosion. That's not going to react it with oxygen. Yeah, it will oxidize and it will blow. Okay, and so you need to do it in discrete steps like this. Okay, that's why we use an electron transport chain. Is that every time we go through a certain complex, we extrude a certain number of protons. And so we need to know a couple of different things. We need to know where those protons are extruded. We know where they're going. They're coming out of the matrix into the intermembrane space. That's not difficult. But which complexes produce that flux of protons? And then what determines the order? There's a very characteristic, the standard reduction potential determines where those people sit in line. Okay? And there's a reason for that. Okay, so here's the picture. This is complex 1, skip 2, 3, 4. Okay, so this is where NAD delivers his electrons. This is called complex 1. And then there's going to be a little mobile carrier. It's got it's made out of lipids. Coenzyme Q, you may have heard of it. Ubiquinone is another name for it. And coenzyme Q is cool because it's isoprene units. So it can never leave the membrane, but it's very mobile. And so it takes all the electrons and moves them over here. Okay? And so you extrude 4 protons in this complex 1. So now you're starting to build up the PNF. Christy's unhappy about that. But so what? Okay. And so you extrude more protons in complex 3, and then we're going to hand off the electrons to somebody we talked about the last lecture, which is cytochrome C. Remember, when cytochrome C is located in the intermitochondrial membrane, it's water soluble. And so it can hop between these two very easily and deliver the electrons here. And if you take a pair of electrons, you'll get 1 water, but actually complex 4 waits for 4 electrons, and then it takes 1 oxygen and makes 2 water molecules. Hence the term chemiosmotic, you're making water. Okay. All right, and so then you take that PMF, that concentration of protons, and we're going to run it through the ATP synthase. So that's the plan. So basically, we're just looking at the stoichiometry. So this is the order, complex 1, coenzyme Q, or ubiquinone, I don't care what you call it. I'll call it coenzyme Q, all right? And then complex 3, so it's 1 to 3, carried by ubiquinone, gives them up to cytochrome C, gives them up to the complex 4. Okay? And the key thing, though, is to know how many are coming through here. This looks like a lot of detail, but this, again, it has weird names, but we're going to call it complex 1. Okay? And this is complex 3, and that's complex 4. So 4 protons are extruded when NAD gives up his electrons to complex 1. That extrudes 4. Coenzyme Q picks up the electrons and moves them to complex 3. In that process, 4 more protons are extruded. So now you've done 4 plus 4, that's 8 electrons. Protons, sorry. So those electrons now, after you've extruded the protons, are going to hop on top of cytochrome C, and he will carry that to complex 4. What happens if cytochrome C leaves the intermitochondrial membrane, leaves the intermembrane space and goes into the cytoplasm? What happens? The cell kills itself. So he does not need to leave, okay? And so when he's functioning in a normal living cell, cytochrome C does not leave the intermembrane space. But it transiently becomes an indirect peripheral membrane protein in 2 different places. When it touches complex 3, when it picks up the electrons, and when it touches complex 4 to deliver the electrons. So it's a soluble protein. It's kind of like ARF that can go both ways, but it doesn't have any retractable landing gear. It just touches that briefly, So it can be a soluble protein or a transient peripheral membrane protein. Okay. And then so the total is 10 protons. Why is that important? So here's the rule. It takes 3 protons to make 1 ATP. So how many ATP can you get out of 10? About-ish. 3, right? because 3 times 3 is 9, and we got 1 left over, we can use that in other ways. So we're going to actually be able to take those 10 protons and pump them through here, and we'll come out with 3 ATP, at least. Okay, so that's the equivalency between protons pumped and ATP produced. That's a good number to remember. Okay. Now, why is complex 1 in that position, and why is this one here and there and there? Okay. So this is sort of the religious symbol of the biochemical world. You know, it's sort of the icon. So this is a Christie standing tall here, and I like it because what it shows you. So here's an arrangement of 1 to 3 to 4. Where does NAD, as slow as he does have electrons, and he's going to give them up, but he can only give it up to complex 2. That's where 2 is. Okay? So NAD gives up to complex 1, but FAD can only, he's a little weaker, can only give up to complex 2. The problem is, being a weaker A&M student, when he hands off those electrons, there's no protons that squirt out. He's weak, okay? But the same number of electrons happen in here and here, so FAD can only extrude 6 protons. So when FAD gives up his pair, the most ATP you're going to get is 2, not 3. That's the difference. Now, say, why is FAD weaker? It can only talk to complex 2, and NADH is stronger and can talk to complex 1. Well, you went to A&M, but that doesn't explain the whole story. So the question is, what explains the order? And this is it. Okay. So this is what's called a nasty sounding name, standard reduction potential. And what happens is it's very negative up here and very positive down here. And if you look at this, you go, okay, there's NAD, and we know he's holding these high energy electrons, so it must have something to do with how NAD handles the electrons. And look at the very bottom, very positive standard reduction potential. That's oxygen. That's the most potent receiver of electrons. So here's the rule, and this determines the order. It is the more negative your standard reduction potential, the better you are as an electron donor. It means you have a terrible affinity for electrons. So the more negative the standard reduction potential, said in a different way, is the more negative your reduction potential is, the weaker your affinity is for electrons. That means you give them up very easily. And here's the other rule. You cannot hand off your electrons to another complex unless that complex or molecule has a more positive standard reduction potential. So the reason that NAD can give off to complex 2 is that his standard reduction potential is more negative than complex 1. And the reason that complex 1 can receive those is his standard reduction potential is more positive than NAD. So that's why they exchange. Look at where FAD is. His standard reduction potential is over here, okay? About minus 0.1. The reason that FAD cannot hand off to complex 1 rather, is that his standard reduction potential is more positive than complex 1. He has too much affinity to let go and give them up to complex 1. He'd have to have a standard reduction potential up here around NAD. But he doesn't, so he hands them off to complex 2, which is down here lower. So you have a more positive standard reduction potential. It means you are a better acceptor than FAD is. So FAD's a good donor in that sense. He's got a very negative reduction potential, and so he hands it off to a more positive reduction potential. So think about that 2 or 3 different ways. What number goes with the stronger affinity for electrons? Which number goes with the better donor? Which number goes with the better acceptor? Those are just get that down. But that's what determines the order. Are there any questions about how that works? It might be better, instead of going between those two terminologies, is just stick with either being a good donor or or having a weak affinity. I don't know which one works better. Yeah.
It wouldn't just be the reduction potentials that determine the order, right? Because then it would just go straight to oxygen.
Right. So there is something about the placement of these things and having subunits, but we're just not. You can look that up if you're really excited about it. Anyway. Okay. So now we know what's being done. And so look at this, all this energy. So extruding those protons creates the proton motive force for Christie, okay? And so you've got, most of it is stored as the membrane potential, minus 160, because every time you push that proton out, you left a really strong negative potential behind, because you didn't send any counter ion with it, right? So that's generating that. And then the pH, you're creating a pH rating of 1. pH 7 outside the matrix is 8, so that's a delta pH of 10-fold. Okay. Now, if you put those two numbers together, it's minus 160 millivolts of electrical power, negative 60 of chemical power, so the total PMF produced of this system that's capable of sustaining is minus 220 millivolts of free energy. That's a lot. Okay. But we have to use it by running it through the ATP synthase, or else it dissipates. Okay. So now we turn to this guy, and we already know a little bit about it. I'll show you this quickly, and then I'm going to show you another picture because this is incorrect. I'll show you most of the stuff is correct, but some of it's not. Okay, so you have two pieces to FO, F1. Remember, FO is the proton channel mainly, and here's a little trick. All these alphanumeric characters like little a, little b, and this is b, and that's c. That means it goes with FO. So just the regular alphabet. The Greek alphabet goes with the F1 subunit complex. So this is the FO, little a, b, c. Greek symbols means it's part of the F1 complex. And they do different things. Okay? So basically you got this thing here, and let me tell you what's wrong with it, and then we'll leave it alone. What's going to happen is protons are going to go into the alpha subunit, this A subunit, and they're going to, this is a proton channel. It's a half channel because it goes in halfway, and then it shoots into the C subunit. There's 10 to 12 of these, and when those electrons move, when those protons move in to the C, it starts to turn. It's a turbine. We even know which direction it turns. It turns clockwise if you're looking outside at this, and I'll show you that. So the protons hop on this, turns, and it revolves, and it lets them back into the A in the other half of the proton channel, and then they squirt into the matrix. So the energy of the proton gradient has been captured in mechanical motion of turning. And so we're going to turn this piston, and this is where the F1 subunit comes in. There's a gamma subunit that you can see sticking down in here. That's like the drive shaft. It's rotating because it's attached to that. And it's also attached to epsilon. So the three subunits that rotate, and you should know this, okay, is the C part, C subunit of the FO complex, the drive shaft, which is the gamma subunit, that's that drive shaft, and the epsilon subunit of the F1. So there's two F1 pieces that are moving and only one piece of the FO complex. What is wrong about this? That little delta doesn't belong there. That's delta. They mislabeled it. The artist mislabeled it. Here's the correct picture. It's not as pretty, but it's a good. So if you're out here in your eye, let's say we're all standing in the intermitochondrial membrane space, the intermembrane space, and that's the intermembrane on that wall. So our eye is up here looking down on this. Which way? So we're looking at a hollow tube that's rotating, and it's going clockwise. That's your frame of reference. We know that it's rotating in that direction. When what happens? When protons are flowing down their electrochemical gradient and when ATP is being synthesized. What if we split ATP? You think it'd rotate backwards? Yeah. If it splits ATP, it'll rotate counterclockwise. And I can prove that. We've done an experiment that way. Okay, so the protons flow in, they flow through this, they spin it, start spinning this. It goes. The protons go back into the A channel, and then they go into the matrix. That's the pathway. A to C to A, okay, matrix. And then the gamma is rotating in the same direction that that's rotating because they're attached to each other, okay? epsilon is stuck on the gamma, so it's rotating too. What's holding all this moving part together, in mechanical engineering term, is called a stator. That's keeping it stable stator. And that is the B subunit of FO. That's important. So it's anchored here with the B so this thing doesn't spin out of control, right? What is it anchored to? It's anchored to the bell pepper. See, that's what I call this the bell pepper. That looks like a bell pepper. Okay? And each little piece of the bell pepper are subunits, and here's the rule. The beta subunits are the ATPs. They're the thing that grabs phosphate and ADP and shoves it together. Beta is the ATP, ACE, and the alpha subunit is an allosteric modulator of the beta subunit. It determines, based on the concentration of ATP and phosphate, how fast this thing should go. The second function of the alpha subunit, besides allosteric regulation of the beta subunit, is the B subunit from FO is anchored onto the alpha subunit. So it's a foothold. It's part of the stator complex. This thing is spinning, okay, so you need to stabilize it by binding to the alpha subunit. And you don't want to have the stator bind to the beta subunit because it's changing shape. Every time this thing rotates, the beta subunits will change shape. Okay. So are there any questions about what's going on here? So that's the order. What we need to do is to do some exercises. So what's happening is let me get some gimmicks out here. So this is ADP. It's actually a flat football. And this is a pink tennis ball, but it's actually phosphate. Okay, so what's happening is the gamma subunit is spinning behind me, and that gamma subunit, it has three beta subunits all on either side in three positions. One here, one standing over here, one standing over here, and the gamma's rotating in the middle. Okay, and what's going to happen is as the gamma turns, it's going to touch the beta subunits in a particular way and make them undergo a conformational change. And so here are the three conformations. It's known as open, loose, closed. Open, loose, closed. Okay. Open is when you're ready to receive the substrates. So when you see me open, you toss me, do it sequentially so I won't look stupid. Okay? So when I go open, pitch. Okay, now open, and then I go loose, and then tight is when I push them together. And that's the catalysis, is they just, if you get them close enough, ADP and phosphate will stick together. Okay? All right, so what I want your groups to do is, and I hope you have at least more than, you have to have at least four. Otherwise, if you don't have four people, you can't do this exercise. So one person needs to stand in the middle, and one person get in front, and then it's 120 degrees, okay? Because every time what's going to happen is, when I go from open, in order to go to loose, what happens is the gamma subunit rotates 120 degrees. And then it's going to go 240, and then it's going to go back to 360. So when I'm standing here in open configuration, the person to the left of you and behind you, it goes, what's the order? It goes open, loose. That person's in the loose configuration when you're in the open. And you're going, the order is going actually counterclockwise relative to the gamma subunit, but that's not important. So this person is open, this person is loose, and the person over here is tight. Okay, and so when you say, so one of you is going to stand outside that ring, because it gets kind of weird if you're in the middle of it, and you say, move, okay? And so you'll go from this conformation to this conformation. And then when you say move again, you go like this. So you go open, loose, tight, and in order to let go of your ATP, you have to move again to the open. So the open conformation has two functions. One is to catch the substrates, and one is to release the ATP. So I want 3 people in your group to be the beta subunits, and one person, you can stand in the middle if you want to be really close, or you can stand outside and just say move. But practice, so you got to get the order right. This person out in front starts with open, loose, tight. Okay. Let's do it. But I'll come on up.